Morning. Good morning. Good morning. Good morning. Welcome. I hope you have recovered from your weekend. I hope it was good and whatever it needed to be for you. Perhaps it involved watching a bunch of baseball and being tremendously excited on one night and less excited another night. Or perhaps you're an LA Dodgers fan or perhaps you're not a baseball fan at all and you're like, what the hell is Smith talking about? Thank you you found nearly all of these words, and with a somewhat Christmas, Halloween, Christmas, the next kind of big holiday, we'd like a red and green with some lights and some tinsel. Let's take those off. Thank you for doing that. It remains for anyone who wants it later on. Let people in from the waiting room, and we get underway. Where have we been? Where are we going? We're still talking about eating. We're still talking about feeding, so I hope you have... actually, today, maybe don't think so much about snacking, because today is about parasites. Like a week ago today, we were talking about deposit and suspension or filter feeders. On Wednesday last week, we started talking about grazing and browsing and hunting and symbioses and some of the incredible symbioses that were revealed the 50 years ago that we discovered and began to appreciate all of the life that is adjacent to these deep sea hydrothermal vents that span our globe, but take up, as you know from your projects, only a very small amount of surface area, and they all become very site-specific, and the tight associations that we see sometimes between members of this symbiotic partnership. Today, we're going to talk about partners again, but we're going to talk about partners that are on a less mutualistic, on a continuum from mutualism to parasitism. We're going to talk about parasitism today, and we're going to go all over, and in fact, more broadly than just the Dunn phylogeny, because we're going to talk about some protozoans. But we're going to talk about this feeding strategy, and if we're going to talk about parasites in general, really we're like so much of a wealth of these exist within the invertebrates, our polyphyletic home group. So we'll talk about protozoan examples, platyhelminth examples, arthropod examples, orthonectids, maybe the phylum that we haven't, maybe other than the gastrotrichs, maybe the phylum that we have spent the least with in our shared time together in 27 and 3700. And we'll end up talking about unionids and Glaucidia larvae, and so mollusks as parasites in a way that perhaps you'll remember back to 1070 and your diversity of life days. So my hopes for this, for you and for us, is the objectives that I would intend for you to take away from this is the capacity to appraise, synthesize, and interpret how parasites are getting their nutrition, thinking about specific examples, how that affects their host behaviorally, evolutionarily, and from an evolutionary perspective, making specific thoughts about how that pertains to the arms race analogy. And we'll talk about that even today. So we talked about symbiosis before, right? We talked about in this first bullet mutualism, so both organisms benefiting from each other. So you've got your Riftia annelids living at the deep sea hydrothermal vent, and they keep a trophosome like a pocket for bacteria, so they've gotten rid of their gut entirely, and they are just subsisting because of the existence that they have, because the bacteria can synthesize from the sulfur-rich water, and the annelid essentially just makes a home for that and benefits from it. And a commensal is slightly intermediate, where one is really, I don't know what the analogy would be, but one benefits, one doesn't. One is really like one's really went into one of them, and the other is like, oh well, you're here, not so bad. But today, we're going to be talking about parasites, where one organism benefits from a close living arrangement at the explicit expense. It is feeding upon the other. And from a life history perspective of that Y diagram of where you can put your energy to, this is something that's landing onto that energy diagram and sucking away energy that otherwise would be used by the host for metabolism, for growth, or for reproduction. Okie dokie. So broadly speaking, and this is a classic view for us together, a classic view of the Dunedall phylogeny with these stamps about where they live in the world, freshwater, marine, aquatic, and then parasitic. And so these red dots indicating kind of where we think or where we see taxa that exhibit this kind of a lifestyle, so that lives inside or on another and causes it harm so that it can feed, so that it can reproduce. So it's like predation. I put it we talk about it in the feeding section or chapter of the course, because it is feeding, but the host is typically much larger than the predator. The prey is much larger than the predator because you're living on or within. And they typically derive nutrition from potentially just from one prey item throughout their life. So it's not in the best interest of this predator that you can see examples. We're going to jump into these examples across the Dunedin. There's like 11 of 34 taxa here, invertebrate taxa, that have red stamps, and we're going to jump across almost half of them, or a little more than half of them today. So from the parasite's perspective, and this comes into the Lewis Carroll diagram that we just saw, it's not in the best interest of the parasite to outright kill the host, because then potentially you've lost your puppet master controls of that host to walk you or swim you or fly you towards the next stage of your life history that you need, whether it's infection or dispersal. So you're going to feed on this organism, but you're not going to kill it, ideally. And our own classification, our anthropocentric view of the world, we classify these parasites based on the effect they have on the host, the lifestyle that they engage in, and the habitat that they live within. So they might be obligatory parasites. They might be facultative parasites, etc. We'll unpack or reintroduce some of those terms into our vocabulary as we go. But this effect on the host, this kind of race between I'm going to live on or inside you and derive my nutrition from you, I'm going to, through time, I'm going to reproduce more efficiently and have more offspring if I can escape that predation event. I'm going to find you out again, and the parasite, going to reproduce more effectively and numerously if I can find you in this new evolutionary space, or this morpho space, or this functional space. And so there's this analogy that we use that grew out of the 20th century, when we were very concerned about, rightly so, about a nuclear arms race between participants that wanted to deter the other side from using their weapons by just more and more would deter, more and more will deter, more and more will deter. And so in this, we take that analogy from Cold War politics in the 20th century to think about the evolution of species involved in a parasitic lifestyle. So species A evolves in an adaptation in response to species B. My wording has been different than I've used on the slide here, because I'm going to use the in response to, but because it's simple, and so visually that's where it sits. And I've spoken the more realistic phrasing, which is, this is not directly in response. This is only in an evolutionary sense is this in response, right? So it's not an organism going, well, I seem to have a bunch of leeches on me today, or a bunch of protozoans swimming around and making cysts in my brain. So I will concentrate hard, and there's an adaptation that gets me away from this. This is not that at all. So the in response, and I know you know this, right? But you are the fortunate few that get to study this in a dedicated fashion and understand it. So this is my aside, to remember to be careful about language, and specific about language, particularly when you're speaking to people who don't necessarily have the privilege of studying this in a dedicated fashion, for whom in response to could very definitely get them to start thinking, well, a Lamarckian thing. It's like, well, I need it, so there it is. All right. So, but through evolutionary time, through the processes of natural selection, escalating adaptations and responding counter-adaptations, And this, of course, this occurs, and this arms race, we can visualize it between predators and prey that don't engage in a parasitic lifestyle and are just simply predators and prey. But it's most perhaps intimately and closely understandable with parasites and their prey, because the parasite really needs to easily infect. That's like such an intimate, an important aspect of their life history. And the host needs to be strong enough to survive parasitism. And so essentially what we have is this positive feedback, or another way to think about it is antagonistic coevolution. So not coevolution in the sense of the mutualist examples from last Wednesday at the deep sea hydrothermal vents. That's coevolution as well between bacteria, the sulfide fixing bacteria and the annelids. But this is in fact antagonistic coevolution. So an escape, tracking it down, escaping again, tracking it down again. And so that's the metaphor of the arms race. Now, the metaphor we can look at graphically, and as you have learned, my real goal for us together is to think about things graphically. So what I would like you to do is to find your annotation tools, if you could, and in thinking about an evolutionary arms race, take a look at this. We've got an x-axis, so we're talking about fossils here. So, and we're talking about mollusk fossils from the Cambrian through the Ordovician and the Silurian, when our Guelph area was the bottom of a deep sea. And what the y-axis is plotting is, pardon me, I'll try and mute if that's going to come up again. I'm pretty sure I didn't capture all of that before it happened, so my apologies. The y-axis is the proportion of this thick and narrowed aperture, the opening of the molluscan shell, which we know from our living with contemporary mollusks. We can think about that from an ecological perspective as a defense against predation. The smaller the door, the less likely that a predator gets in. So find your annotation tools, take a second, and plot for me the trend you would expect to see in the proportion of this thickened and narrowed aperture, this adaptation against predation. What should it do if we to see evidence of this evolutionary arms race? What's the trend we should see? Cool. Awesome. Now, some of you are putting a linear change. Some of you are putting some inflection points. That's all great. If you've got any specific hypotheses about why they're... I got almost all the way through the course without doing that. Some of you are putting a linear trend. Some of you are putting inflection points, or if you've got any thoughts about why one or the other, throw that into the chat if you would, while I search for my white erase marker. And thank you for that, because you're pretty much exactly right. It is an increase that we see a real change and evidence of this adaptation, counter-adaptation, that sometime at the end Permian, so after the Permian extinction, we see a real change in the proportion of this defense against predation. Now, so what's happened here at the end Permian? The world changed dramatically. Well, for one, up to this point, up to the Permian, mollusks existed. Like, we have molluscan fossils in the Ordovician period and in the Silurian period, but they were much less numerous than the other kind of almost or bivalve looking phylum, in this case the brachiopods. So we see a real change in the diversity and of mollusks that happens there. That's not in the graph, but that's just what happened. But we also see this characteristic change that suggests as diversity increases, there was more opportunity for different kinds of predators. So we see the advent here at the end Permian of this arms race between mollusks and their predators. Now we can look for coevolutionary evidence of coevolutionary antagonistic coevolutionary arms races that go in the opposite direction. So here we've got, with roughly the same x-axis, a little bit truncated time period, but we see the decline coincident with the increase in the I'm better defended against predator. We see the decline in loosely attached forms that are pretty poorly defended, and that they're just kind of like sessile and sedentary and uncemented gastropods through time. And so this arms race analogy, you can make it. It's kind of it's an auditory, it's a verbal back of the napkin. It's like this, an analogy, but we can also express it graphically when we have data that we can look at in a clever fashion. Comme ça. All right, there is my friend Caramel. I don't see any new questions or thoughts in the chat, so we will continue to go through our zooming through the amuse-bouche of the parasitic lifestyle to think about habitat. Now, habitat here, we're not talking about explicitly about marine parasites or aquatic parasites, but let's talk about the environment from the parasite's perspective. Those parasites that live inside the host or on top of the host. So the endoparasite or the ectoparasite. And you know a zillion examples of each of these from your experience as a human and as your training as a biologist. So from an endoparasitic, you can think about like toxoplasma and tapeworm and things like that. Exoparasites, we've got fleas and ticks and lice and leeches and monogenean trematodes, all those kinds of things. And we'll unpack some of those today, or as a reminder. Now, you can have another description that you would use in a parasitic, if you're a parasitologist, is the classification endo, so by habitat, endo, ecto, classification by the effect on the host. Are they non-pathogenic? Do they not cause a disease? Is there very little expression or effect of the parasite to our eyes on the outside? Or is it one that causes a disease, like hookworm or entamoebas, things like that? Now, finally, an effect by lifestyle, a diversity of phrases here that you can be, we talked a little bit about already, a parasite that can be parasitic, but it can actually, if required to, can complete its life cycle without a host, or an obligatory parasite that requires, so like some of the wasps and bee examples. And then a permanent parasite, like the tapeworms, things like that. And then a temporary parasite like your leeches, or other kind of blood or hemolymph feeding organisms. So how do parasites feed? So what they do in the shadows, those vampires, those are essentially parasites, right? Because they get food from the host or at the expense of the host. So many are actively just feeding on the blood. And by attaching to the host, or they can, if they're an ectoparasite, they can feed at any time. The feeding mechanism, we commonly think of them, if we're going to break them out in terms of categories that we introduced last week, they're hunters. Right? That they're searching and they're responding to cues, environmental cues, whether they're chemoreceptory or movement, so from light, those kinds of things. And by this hunting down, a leech responding to movement or a chemical in the water, swimming upstream of that towards that disturbance or towards that chemical cue and attaching to its Guillermo, it may kill the host or it may not. And part of that is when, from an ecological, the kind of intersection of ecological descriptions and observations to evolutionary trends, of course, these relationships are being broken and built, and that arms race is progressing through time. And when we enter the arena to kind of look around and see who's eating whom, we may be observing a relationship that's comparatively established or comparatively new. And one of the aspects we would make here is what the point that the bottom two bullets are making, which is building on a point that I made earlier, which is the parasites is not really to their advantage to kill the host, but if the association is a novel one, or potentially if the host range is broad and there are some more ideal or less ideal hosts, then you can have unintended or less than ideal outcomes, i.e. The host could die. So how do parasites reproduce? And that is that's not just a course, that's a degree, and because on its own, and potentially postgraduate work as well, because there are so many species of animal that are not parasitic, and then there are so many species of parasitic animal upon those, and many of those are specialized, i.e. one species of parasite for one species of host. There are generalists as well, but there are a multitude of responses, nearly as many as cases that we see in the world. There is beauty in this view of life with its varied forms, Darwin says at the end of The Origin of Species. And so much of that beauty is in the variety of ways that there are things out there trying to eat and kill you. So we'll think about some common elements. One of them is that they have and when we were talking about life history strategies, that we talked about sexual and asexual reproduction. And we talked about so parasites often can switch between sexual and asexual reproduction within different hosts or different life history stages or different intermediate hosts. And of course, advantages of sexual reproduction include increasing diversity, and then asexual reproduction can be fast and can occur in the isolated kind of meta population, the little bulb of the goo that you're living in moving through the world. Some hybrids or some parasites can hybridize very rapidly, and so that retention of the ancestral trait for two species to be able to interbreed and produce fertile offspring is maintained for potentially a longer period of time. And so looking at this schistosome worm, we've got, and you'll see some of these, and you've seen some of these CDC life history diagrams before, but we have adults existing in its host that are then via feces, they are moved out, they move eggs out into the environment, and then they move into an egg into different larval stages, like the Myricecadia larva here of this schistosome worm that then searches out some snails, crawls inside the snail, and begins to move through different kinds until it looks, in the end, this Sicari larvae that burrows then out of the snail to invent its second host, which could be a bird, and it could be another snail, it could be a fish, depending on the species, and then all in all, we keep moving along and along. Oh yeah, I like that. I'll heart that as well. I just looked down. I'll give it an ant because I don't have my heart. But maybe we're all parasites in our own way. This is going to become very circular. Dude, I think we're all parasites. I definitely have felt parasitic at various parts in my life. And less so in others. So let's talk about, one of the goals that I've had throughout our time together in this course is to advocate for things that we don't think about often, and I'm going to change so de-emphasizing the taxa that are important to our own species. But let's change Smith's game plan a little bit here and just spend some time talking about some human parasites that fall, I would say, into three main groups. We have protozoan parasites like this Giardia, which would be that kind of a great costume, I think, for Halloween at the end of the week. Parasitic worms and ectoparasites. And the worms is in quotes, the Tribbiani quotes, remember, because worms here is used by parasitologists to mean a whole number of different phyla. Multiple phylum equals phyla. So let's dive in first of all to protozoans. So that was the first like almost a month, say 2 or 3 weeks of 2700, right? Was our acknowledgement that when I was in your place, there would have been a course entirely of protozoology at the University of Guelph. And with retirements and budgets, savings and decisions, effective leadership, we don't have that course anymore. And so this is the course, or that was the course where we still talk about it. And because these two courses are associated, we'll continue to talk about protozoan examples here. So These microscopic single-celled organisms, much greater phylogenetic diversity than just the metazoan invertebrates that we focus on. And of course, they can be free-living or parasitic. They can multiply easily in humans. And we have a number of different, very significant infections that can develop from very few organisms. So there's a couple of diagrams here. This is the apicomplexan Toxoplasma gondii. We'll talk about Toxoplasma in a second. This is Giardia here. This is a slide, an SEM that's looking at some pig duodenum, I think, and nestled into all those microvilli inside the duodenum are all of these little swimmers. This is a pig that was having a very bad day because its guts are full of Giardia. And that fecal oral route, that typical transmission, so this Giardia is probably inspiring its host to then begin voiding from either end of the alimentary canal in a very unpleasant fashion. That fecal to oral transmission is a fantastic way to spread your eggs out into the environment, and also then to spread, to take them up, especially if you're consuming water that has not had the benefit of having different levels of filtration. So let's return to thinking about Toxoplasma gondii, because it is one that, although we as humans are not the definitive host, it certainly infects us. And as you remember, you might remember from 2700, it's a parasite, a protozoan parasite, that some estimates have its rate of human infection as ranked or estimated as high as 90%. 90% of people in the world have potentially these little cysts inside of their brain that are caused by protozoan parasites. So the definitive host for the Toxoplasma gondii is, of course, the cat, some cat species. And the cat, then inside of the cat, we see sexual reproduction, and then we see the release of different cysts out into the environment that then infect other taxa. Other taxa, ideally, where different parts of the life history stage can take place and they can multiply. This is kind of like a bootstrapping stage, so numerically they can increase here in their intended intermediate hosts. Prey items of the cat, rodents, birds, that kind of thing. Now, often these tachyzooites, and again, don't worry about the details of the descriptive details of the different larval stages. If I'm not ever going to ask you a gotcha question about what is the name of this, because that's a thing that you can look up, and who cares? You can look it up. I don't need you to memorize those kinds of things. But I want you to think about, so what do those cysts do in the intermediate hosts, either the intended or the unintended hosts? Now, one of the things that we know from some careful work in the late 20th and early 21st century is that most of those cysts seem to increase risky behavior. This is a rat that the cat looked at as a meal, as a predator would look at a prey, and then this rat said, "Not a chance, I'll fight you all," and chases it away. So this is potentially a rat that has many of these Toxoplasma associated cysts in its brain that are increasing its risky behavior. So we see the cysts go out into the unintended or intended hosts, and they can live, and these are kind of some overarching, there's the specificity of the Toxoplasma example, but there are some generalized extensions here. One of them is manipulation of host behavior to further the movement into the next generation. How does the risky behavior of that rat further the movement of the Toxoplasma? Well, eventually that cat's going to turn around and eat the rat. And even though it's looking like, oh, that's an increase in risky behavior, well, it's making it much more likely that that rat is going to become metabolically part of the cat, and so therefore will part of it facilitate the transmission of the Toxoplasma. So that's one of the, and that's a generalized strategy for parasites. Another is that we talked about this kind of asexual sexual reproduction, but also voiding your eggs into the environment and making them little time capsules. Again, so much of parasite biology, if you think about aliens, any of the Aliens Earth, any of that kind of, those are all really parasitology textbooks written up in a sci-fi kind of environment. So oocytes that can survive in moist soil for 2 years and maybe even longer. They can be moved around by ocean currents. They can affect their intended hosts and their unintended hosts. So sexual reproduction, in this case, happening in the cat, asexual reproduction happening in the intended or unintended hosts. Now, parasitic worms, multiple phyla. So we've got platyhelminth, acanthocephalans, nematodes, all of these things. These are not, none of them are true worms. None of those are annelids, but none of them are, but they're worm-like. And so the Schistosomia that we saw earlier, here's another example here. Swimmer's itch, but also some nastier, many other nasty diseases that affect our own species. So Schistosoma masoni. So millions of humans affected, especially in warm regions that work in close contact and live in close contact with water, where that water is not cleaned, rice paddies, river sites, that kind of a thing. And these, when infecting the human, can live inside the liver for up to 25 years. And consider how easy it is to spread and to maintain a population once it's been installed or arrived in an area, when you can, if your one little organism sitting inside of your liver can produce 300 eggs a day for that 25 years. And so voiding it out into the water and then sharing it amongst the snail population and the rest of the vertebrate population in the area. So human ectoparasites, we've all just finished summer. Many of you were thinking about them. One of the, of course, the classic ones that has taken much more attention in the last several years, last kind of couple of decades in this part. If you're from Ontario, if you're from Southern Ontario, you know that ticks have arrived. We didn't used to see them in Ontario, kind of north or anywhere. If you couldn't see Lake Erie, you never really saw a tick. But now they're all over the place. And we're concerned about them because of other diseases that are associated with and vectored by the ticks. And so, broadly speaking, a human ectoparasite like this would include kind of all sorts of blood-sucking arthropods, like your black flies or your mosquitoes. But it's maybe more narrowly, if you wanted to make that a more narrow definition, it's ones that feed for a longer period of time. So less so at one end, like the fast blood feeders like the black flies and the deer flies and the mosquitoes, and more of the long-term ones like your ticks, your fleas, your lice, and your mites. And we talked a bunch about ticks last year. But just Lyme disease, we know it's a disease. It's an emerging health threat in Canada, in Ontario in particular, that's going to affect the rest of your days, anyhow, as this main tick vector, Ixodes scapularis, becomes entrenched in this part of the world and kind of oscillates up and down with its intended host rodent populations. And this is an analysis that was done early pandemic, just about the rates of Lyme disease in and around Ottawa, and they were correlated really from a public health perspective, a non-surprising way, with several sociodemographic variables, that the more affluent areas had less incidences of Lyme, and the less affluent areas had have less time and less resources, and there were more incidences of Lyme there. And so this is, of course, this is parasitology intersecting with sociology and epidemiology. Other ectoparasites include some of these amazing critters that you've seen in your lab, but also if you go up to Algonquin Park, or if you're from that part of the world, but if you take the ecology field course in and around Algonquin Park, maybe you'll meet some of the relatives and the descendants of these leeches that were involved at the Wildlife Research Station in Algonquin Park in one of the last iterations of the course that I taught. Now you've got, and you know, the stories about your leeches, your lab leeches, that came about in 2019, when there was a man from Niagara Falls who was fined or was caught at Pearson flying into Canada with a suitcase full of leeches, trying to smuggle nearly 5,000 live medicinal luggages in his carry-on baggage around this time of year in 2019. Now, he was caught, and these are some of the border control officers are transferring some of the leeches from their intermediate life while the trial was going on, but this is them arriving at their lab life now. Sherry Hinks was responsible for getting in touch with some of the ROM, the Royal Ontario Museum experts who were involved in that trial, identifying the leeches, which species there were, and then saying, well, once you're done with them, can we give them a home in the lab? And so there they are. So the leeches, it was a dog that identified the bag, and then some of the leeches were identified. Now, identifying leeches is kind of a challenge, and so what they had to do was use genetic methods to identify the leech itself, that it was this particular, it was not the medicinalis, which is the more commonly, more widely distributed and used medicinal leech, but it was verbena. And so you can see there's a little bit of some morphological differences there, but it was genetics and barcoding of the leech tissue and the host tissue that showed where they were, what they had been eating, that permitted the trial to proceed. Now, when some of these leeches die, some former students of this class who worked in my lab and were working on the scanning electron microscope, we took some of those leeches, and the challenge was for Mohammed to show us what their teeth look like, what they're feeding, how did they intersect with the host. And so this is a mandible from one of the leeches at a pretty large scale. You can see the serrations. So let's zoom in and take a look. And this is the teeth of a leech. This is one mandible. So you can see they've got these teeth that would be essentially chainsawing or saw blading through the tissue to begin to release their hyoidinin, which is the anticoagulant that inspires our blood to just continue to flow, and their meal to continue to grow and to continue to grow. And I just can't, I know sometimes I stop and just kind of like stare at some of these SEMs, but this is gorgeous. And that's, I mean, look at the reticulum, that's a chainsaw. It's crazy. So if you're going to go out and design a better and more effective chainsaw potentially, and go on Dragon's Den eventually, take some SEMs, come to my lab, work on some SEMs, get some new vision of leech teeth, and you'll probably get a more effective chainsaw. So our final kind of moving through some of the last examples, we've got nematophorans, or these horsehair worms. These live inside of the guts of terrestrial arthropods, like this unfortunate cricket, that then move their host, induce the host to move towards water to begin where they can jump out and move and find their next mate inside of the water source. So this is a long Gordian worm. That activation or the inspiration that it provides to the host, to the definitive host, is unknown. So we just saw this mass emerging from the definitive host into water, where they live as adults. They create some eggs. Eggs transform into larvae that sometimes find a paratenic or an intermediate host that moves them around and takes them out of the water and eventually kind of finishes this life cycle. But this is, again, we know next to nothing. This is very much the Jon Snow element of biology, that we know nothing, Jon Snow. The diversity of these Gordian worms, we used to think very commonly in North America, there were several species that were widely distributed and could eat practically anything. And partially that was because morphologically, they're what a taxonomist would call depauperate. They don't have a lot of characters. These horsehair worms, when looked at under an SEM, they look like a long bit of worm, and there's very little for us that we use using our visual biases to differentiate. But what we found with the advent of genetics is that there are many more species, even just in recently deglaciated area like northern North America, than we had previously thought. So there are estimates of the specificity, the host specificity of these and the geographic ranges. It's changing all the time, and it's generally changing in the direction of more host specific. So that's altering our total estimate of diversity of the things on the planet, and also providing many more questions about host manipulation, different strategies, different hosts being inspired to move to different places. In this case, it was just the advent of water in a petri dish to this unfortunate cricket. So we leave the arthropod, or the interaction between the arthropod and the nematophoran, and let's think about the orthonectids, because nobody's ever thought about orthonectids before. Because basically, they look like this tiny little SEM here in the bottom corner, like a Dr. Seuss puppet. They're gonochoric. This is a female full of eggs. This is a male, little torpedo, kind of dancing around, maybe at Mardi Gras, filled with sperm. But it's a worm-like thing that for a while was characterized as a tiny, very reduced annelid that had none of the other characteristic of annelids, but we didn't know where to put it. But it's worm-like with a layer of ciliated cells on the outside. And they swim around in the body of the host. They're not attached to anything. This is just, they're living in inner space. Their outer space is your inner space, or the inner space of the, not of you, but of marine invertebrates, because that's where they're living their gonochoristic lifestyle. But it's very simplified, super small. This is maybe 100 cells is the organism. But then, behaviorally, really complex, and we know next to nothing about it. We know that this plasmodium seems to exist within this plasmodium stage within the host here on the right-hand side of the clock face, that tends to castrate the host, prevents reproduction of the host. So there is a negative effect. There is a manipulation of the host. We don't know what or why that happens. And we don't know the number of cases that it happens. And then there's this whole stage where it lives in the plankton that, again, we know nothing about. Now, in this case, the sexual reproduction is happening outside the host instead of some of the other examples. So in this case, it's happening out in the plankton. Then the larva tends to infect, maybe it's a young annelid or an echinoderm, but then we've got repeated kind of asexual reproduction and amplification. So its phylogenetic position is unknown. You'll see it appearing in the Dunn phylogeny as a polytomy. It might be closely associated with another very reduced group, the gastrotrichs. Remember the ones that were feeding the filter feeders upon the filter feeders? But it might not be. And its life outside the host, we know practically nothing. Now, some more arthropod examples here as we work towards the end that I'm going to guess you're familiar with it because of the number of bee researchers and bee experts that we have around the faculty here or around the campus at the University of Guelph. So this is the mite, Varroa destructor, a parasitic mite that lives in association with different honeybee species. Now what happens here, just briefly, because I'm going to presume, and tell me in the chat if you want me to expand, but we have a female mite that lives on, it's living its phoretic life. It's basically living on adult bees out here, and it can spread its own, it can spread disease, it can disorient, it's taking away hemolymph from the bee itself, from the adult bee. But then a lot of the really, from a beekeeper perspective, the insidious damage is caused when that infected adult enters the colony and then lays its eggs on larvae before the bee larvae are capped in the colony, and then it spreads, it eats, it consumes, it weakens bees by consuming the fat bodies. It can act as a vector for at least 5 other viruses, and a significant infestation can not just reduce the vigor of a colony, but they can kill that colony. So there we've got a mite living on an adult, and then a mite on a larva. Now, some of the interesting, to me, biology, interspecific relationships about this is that Varroa mites were first reported in Java in and around Indonesia in 1904, so 120 years ago, on a different, not on the honeybee, but on a Apis cerana. And it was relatively recently, it was in the 1950s, in the middle of the 20th century, when we first started to document host switches to Apis mellifera, the honeybee, the globally distributed kind of chicken of the insect world. And that happened in Japan and Hong Kong. And then, in a relatively short period of time, by the 1960s, we entered the picture and we start moving it around the world by illegal and legal honeybee trades. And it's hitchhiking on ships. So we're now essentially responsible for the movement of this parasite all over the world because we've moved its host all over the world. And so it exists basically in all regions where humans manage honeybees. There's some remote islands and maybe the extreme north, but I'm going to suggest that with the continuing climate crisis both of those things are going to change. Another example, a new phylum, but with trematodes. So parasitic platyhelminth flatworms and how they intersect with mollusks. So the primary host is birds. The intermediate host is the land snail or snails, terrestrial mollusks, terrestrial gastropods. They infest the snail, and then their brood sacs are this. So this is the antenna, the eye stalk, call it, of the gastropod. So the trematode, once it's infected, this mollusk migrates in the body up into that eye stalk and essentially pulses, and is literally like a neon sign, figuratively, like a neon sign for the birds. Then, so the snail, the gastropod, can no longer hide as effectively and is then much more easily consumed and moved, and the life history of the parasite moves along. So another example of the host's behavior being manipulated once it's parasitized. So it alters the snail's behavior to make them more likely. So not just making them more visible, but it makes them more mobile and makes them move to more higher up points where they might have avoided otherwise, because those are the most likely to be dehydrated or to have water loss stress up there. And you could, and again, we can talk about this kind of verbally, but you can look at it graphically as well. So here, what we're looking at is infection of different snails. So the prevalence of the parasite here on the top graph, we've got the infestation of the trematode increasing very dramatically away from the water's edge. And here we're looking at mapped out the snails that are not infected versus the snails that are infected in terms of where they sit in space. And so this is graphically looking at that pulsing eye stalk to say this as an example of this adapted behavior, of this altered behavior of the host. What does it mean for the snail to be taken away farther from the water's edge? Well, it means essentially that parasite has made it much more likely that you're going to be consumed by a bird, and then you're going to fly away. Finally, Glaucidia. This is such a gorgeous story. So you live in this part of Eastern North America in the last couple of minutes of this lecture in an area that used to have most of the diversity of unionted mussels in different river systems up and down the Eastern North America. And of course, you know this from 2700 and 1070 before it, that there are these tight host-specific relationships between the mollusks and the Glaucidia larvae, these little Pac-Men that they release that go out and parasitize different fish species that either are inspired or to bring the host closer, and that they live as blood feeders, blood-feeding mollusks, blood-feeding bivalves on the gills of the fish. A heavy infestation can make respiration difficult, and then there's different adaptations associated with different species-specific relationships. So there's a zoom in, a kind of a beautiful line diagram from your text last year looking at that Glaucidia larva with the attachment thread and the sensory tufts that say when to close. And then you can see the mollusk kind of shooting them out here, shooting them out, and filling the water with all of these different larval forms. And of course, then there are the species that have adaptations of the mantle that look like prey items for the particular species of fish that they wish to infect. And so goosh, that release that you just saw, that was the Glaucidia that were released and then glam on to the gills of the intended species. And so we see some taxa, this is a co-evolutionary, a co-phylogenetic relationship. So we've got fish phylogenetic, fish phylogeny on the right, and mollusk phylogeny on the right, on the left. And in some cases, you see really specific tip-to-tip relationships where closely related species of mussel are infecting species of mussels are using very specific species of hosts. So summing this up at the end, some of the lineages we've seen were exclusively parasitic, some are not. There's a whole bunch of this, so even though it's 11 of 35, it's very widely distributed, but we're learning still more and more things about this. And one of the things that we've learned relatively recently is that even the ctenophores that are lacking this red dot, there are examples of parasitic ctenophores that are parasitic to salps or tunicates, are kind of close relatives. And in fact, then parasites upon parasites, this was a species described a couple of years ago where there are parasitic annelids that live on ctenophores. Unknown biology, other than that they exist. And you can see that wrinkling, furrowed brow of the concerned ctenophore. All right. Like a Concorde jet view through the strategies of parasitism that we see within the multiple phyla that make up the purview of this course. Where are we going to go next week? Where are we going to go for the rest of the week? In the lab, you're back. You're still thinking about your project because your peer review assignment is due at the end of the week. Also, at the end of the week, you'll have your next case study released. The lab is going to be about cricket, snails, and crayfish. And on Wednesday, we're going to begin kind of a 3-lecture session on ways of perceiving the world. And we're going to start out with photoreception and think about eyeballs and light. So until then, take care of yourself, take care of others around you. I'm going to stop recording and hang out to see if there's any questions.